Well, good afternoon, everyone. It gives me great pleasure to introduce Dr. Jose Julio Ortega from the Spanish Research Council. So he's a senior researcher at the Spanish Research Council and located at the Institute for Natural Resources and Agrobiology in Seville. So I've met Dr. Ortega over the last 10 years or so. He did his PhD in microbiology from the University of Seville, and then went on to postdocs at the University of Amsterdam and Cornell University. And then over the years, he's taken sort of short stays at various universities, at MIT, at the Swiss Federal Institute for Aquatic Sciences and Ecology, and you were at Oxford as well more recently, right? So the area of his work is sort of bioavailability of organic pollutants from a perspective of remediation of contaminated groundwater and contaminated sites. But he's heavily also related to policy aspects that touch on the issue of safe chemicals. So Dr. Ortega is past president of the Society for Environmental Toxicology and Chemistry, the European chapter, and he's involved in a whole bunch of other European organizations on legislation for safe chemicals, like the European Food Safety Authority, for example, and the European Chemical Industry Council, and so on and so forth. So very plugged into the regulatory world. He's also associate editor of Science for the Total Environment and critical reviews in environmental science and technology. So very prolific researcher. It's a great pleasure to have you here to share your work with us. And so with that, Jose Julio, I'll turn the floor over to you.
Thank you, Surpassing. Thank you very much. First of all, I am honored by this invitation by Professor Goschal and the TISED Institute to give this talk today and to share with you all our ideas on bioavailability research on organic chemicals. So I will divide my talk in two parts. One more oriented to the basic research we do on engineering components related to biodegradation and bioremediation, and the second part more oriented to regulatory aspects and also how to measure bioavailability and also having a view of what is happening in the regulatory arena, but mainly as a very personal view, as I have not been involved directly in these regulations, but I will show you our ideas how bioavailability can also provide input to these different policies. So I will start with some general concepts about bioavailability. Some time ago, we did a communication exercise that we published in Environmental Science and Technology with different stakeholders on interrogating us why such a big research of bioavailability of organic pollutants during the last 20, 30 years had not found the right way into regulation. So we discussed with people from the industry, from the regulatory agencies like ECHA, European Chemical Agency, how we could facilitate this knowledge to be implemented in regulation. Sometimes you have to make some assumptions, some simplifications that are needed for a regulatory arena. So I will go later to that. No? But also was a good chance to look at what was the current state of the art in bioavailability. So looking at the different bioavailability processes. No? Bioavailability can be composed by different processes coming from the exchange from the polluted soil aggregate or sediment with the adjacent water, reaching or not equilibrium. Also, this released contaminant needs to be transported through the biological membrane through process B or also directly from the sediment surface to the biological membrane by the direct uptake. Later on, once in the organism, there is a whole set of different biological mechanisms depending on the biological entity we are treating. Not only biodegradation, but also toxic effect, etc. Okay, we will go later on what is happening with this toxicological view with biodegradation. So in the literature, we saw different ways of defining bioavailability, and this has been also part of the problem in going to regulation, because we as scientists like to complicate a bit things, which is nice. We understand the nature better and so on, but when you go to the regulator, you have to have clear messages. But in general, they are quite useful for us in understanding bioavailability processes. We can go to the work by Trottier and collaborators of describing these processes. Later on, Kirk Semple from Lancaster differentiated between bioavailability and bioaccessibility, incorporating some of the research also on metal regulation, which is including the time coordinate into bioavailability exposure by having this bioaccessibility concept. It's useful especially also in biodegradation research. Also, Reichenberg and Meyer later on differentiated between bioaccessibility and chemical activity, which is also a very useful concept in describing how hot can be a chemical for biological interactions, having this chemical activity concept. All these concepts and definitions have been the base of methods for measuring bioavailability, like T-axis desorption, passive sampling, etc., that I will go through that in the second part of my talk. Okay. So going into biodegradation, which is our subject today, we want to destroy chemicals with microbes, and we have to help them with bioremediation, but that we have to face that not always that a biodegradable chemical will automatically be biodegraded in any environment. So there will be some persistence if we have toxicity, for sure. In the size, we have ecological constraints like low levels of nutrients, electron acceptors. We have low concentrations of pollutants below the thresholds that microorganisms need to be exposed to start to work. And very important, they will not be degraded if they are not bioavailable. They are there, and the microorganisms are very close, but they are not able to meet, and then we have really a bioavailability constriction. So how to attack this from the microbial point of view? So we can see that, for example, once a pollutant is released into the environment, we will see a decline in total concentration of the pollutant with time, and will be automatically an increase in the what we call the recalcitrant fraction, the really fraction which is hard to degrade. This will happen always. A chemical is released into a given environment spontaneously, not really subject to bioremediation or any remediation treatment, but any release of chemicals, and therefore we may have a fraction of the chemicals that resist in soil, even are hardly extractable by organic solvents, so we cannot really detect them. This is the non-available fraction. Okay, so now that we go to bioremediation, so we like to speed this process to and stimulate the organism, and we may have this situation in which I will go back to that later on. So we may have not considering the total concentration of the chemical, but the risks that are given the chemical. Okay, sometimes the risk when you treat a soil with bioremediation, you stimulate bacteria. sometimes even the risk can be initially increased due to the production of these metabolites of the chemicals that even when the parent chemical disappear following the previous figure, but the risk is increasing because of this metabolism of the parent pollutants. So we want in our research really to find way that their risk is decreased, and also that we stimulate the bacteria, we stimulate the microbial population, but at the same time keeping the risk low. And there is a second phase in which the risks are declining, bioremediation seems to be working, but still is not enough for regulatory demands. So there is a sort of plateau at the end of this process that also need special measures that we will try to find engineering components that help us really to go down even at this later stage in risk. Not in the total concentration, but in the risk, and there we find bioavailability. Okay? So that's the more or less the conceptual framework that we try to run in our projects, in our research projects, really try to find ways to do this type of actions in the two different scenarios, high risk and low risk. So coming back to this ecotoxicological view of bioavailability, these bioavailability processes, okay? Now you will see a change from the previous slide. We consider here specifically biodegradation. Okay? So how we can act on these bioavailability processes, phase exchange, transport, direct uptake. We think about microorganisms only in terms of biochemical capacities of pathways, of how many chemicals they can degrade, how many enzymes they have, but microorganisms can do more things, can move, can attach, can synthesize solubilizing components that have an impact on these bioavailability processes, and this has been the major motivation for our research. So my group has investigated really how, for example, how to operate on this released chemical, on this transport mechanism related to the bioavailability, try really to understand how even microbes move through, for example, chemotaxis, a specific mechanism detecting chemical gradients in the soil, pour water, and then try to really operate on that to enhance bioavailability, to enhance biodegradation. Also, we can look at the direct uptake at the interface, promoting attachment to the interface of oil drops or soil aggregates. And there, they are able to take the chemical directly at the site of pollution. So in this way, we have found also ways of operating through this also engineering component of attachment, really how to improve bioremediation. And perhaps the best known mechanism to operate on the phase exchange by producing biosurfactants, for example, or applying biosurfactants also for enhanced biodegradation. So we have prospected ways of, under a low-risk situation, really this is very important that we need to keep in mind, how we should enhance the phase exchange to reduce risk. Doesn't make change to enhance the risk, because you can solubilize the chemicals and create a problem. So we have really to modulate these engineering components really to do the right job in the right situation. So I will give you a few examples of our work and how we treat that experimentally, ranging from model systems that we are controlling in the laboratory with well-known conditions, to more applied situations, for example, in polluted soils, that we treat the soils in different ways. Okay, so these are the type of experiments I will show you. So first, how we can mobilize the bacteria. So it's a very low risk strategy. you don't mobilize the chemicals, you mobilize the bacteria, unless they are pathogenic. Well, we don't want that. But this is quite a low-risk strategy, really, to move bacteria to the pollutant source. Here, for example, you can see a typical reaction of chemotaxis by soil strains, Pseudomonas putida, to phenanthrene, which is a three-ringed chemical, three-ring polycyclic aromatic hydrocarbon. There you can see really that motile bacteria, which are chemotactic to a specific pollutant like phenanthrene, can mobilize, this is in a suspension, a bacteria suspension, a very significant speed. When a phenanthrene concentration of 0.65 micrograms per milliliter, the speed of the bacterial front, when they are exposed to this gradient, can be up to 1 millimeter per minute. So taking the size of a bacterium, which is about 1 micron, you can make the calculation that this could be for a Ferrari car running at 300 kilometers per hour. No? So this could be like this situation, showing you that this is a very significant mobilizing process. Has been very well studied in biochemical, in biomedical science, basic microbiology, but in environmental microbiology there was something also to look at it, and also in connection to bioavailability. Okay, you may say that, well, this is just in bacteria suspension, what happens with soil, no? And we really want to know that this is not the ideal situation for a car. We really need to see what is happening in a porous environment, in a soil, in a sediment, that bacteria don't have so easy to move around. So this was the main motivation, and trying to really understand how we can modulate this mobilizing mechanism in connection to bioavailability of the organic chemicals. And well, the modulation can involve direct chemotaxis to the pollutant, but also you can use chemotaxis to other substances that are typically in nature, like dissolved organic carbon, root exudates, that trigger the chemotactic response, and you can use that to enhance the transport of bacteria to the pollutant source. And this is what is shown in this experiment, where we used root exudates produced in vitro by sunflower plants, and we tested how this chemotactic reaction, when bacteria are exposed to this chemical, they change enormously the motility mechanism, preventing the attachment to the porous surface, and then enhancing the bacterial transport in this model system. In this case was a model breakthrough column system. CC0 values are the concentration of bacteria in the outlays of these typical breakthrough experiments. If you do transport column experiment with soil, you may be familiar with that. And in the graph B, we show very clearly that only the root exudates with a very clear chemotactic reaction was enhancing this transport through this change motility. So this was one scenario in porous system through column system, and we wanted also to know what is happening also when the pores are even so close to the bacterial size. And this was a research we did in collaboration with Professor Ghoshal, where we use a bioreactor designed by him, where really through a membrane you can control very well the size of the pores, where the bacterial tactic response can be monitored, and then we can really interrogate what is happening also in this difficult scenario of so narrow channels for bacterial mobilization. So we got also nice reaction, a nice view that this may happen is so small pores, like 5 microns, as compared to 1 micron of the bacterial cell. And the chemotactic reaction, as you can see in the graph B down, the mobilization of bacteria are very clearly detected as compared to the control without any chemoeffector triggering the tactic reaction. These values means that, for example, with salicylate, which is a chemoeffector, very strong chemoeffector, and intermediate of the naphthalene metabolism, this bacterium is able to degrade naphthalene. We can see a homogenization of the bacterial cells through at both sides of the membrane, which means that the mobilization is very clearly detected. Although we could see that at 3 microns there was a sort of unexpected lack of transport, which was a surprise because the bacterium is of 1 micron and would be expected really to go through the membrane. So we attribute that also to a difficulty of the sensing mechanism of this strain in such a narrow space. Bacteria just move around. It's like the eyes to see the environment, and where they are in such a narrow space, they are sort of blind for these chemical effectors. Although theoretically, physically, they could go through, the chemotaxis reaction cannot occur in these narrow pores. We excluded any kind of artifacts, like could be like the closing the pores to these massive positional cells. So we really attributed that to the lack of chemotactic sensing. So this was the idea, no? That really that the bacteria in such a narrow space they can go through, but there are still some restrictions involving detection mechanism of the chemical gradient. So still good news. In such a difficult situation, we can find ways of helping bacteria to access to eventual polluted sites that are theoretically not accessible to them, and they can enhance biodegradation. So the second process was also enhancing attachment. So we could figure out ways of promoting the growth of attached bacteria by growing biofilms to the oil drops and etc. And then there, where they are so close to the interface, to that specific gradients, then they can just enhance biodegradation. And the one example I wanted to show you is on this, on how we can combine different organisms and then help really with this colonization of the interface. In this specific example, we wanted to test how fungal mycelia that are forming networks around the interface can help bacteria to colonize the interface, and there, when they are growing together, then bacteria can do better job than when they just suspending in the aqueous phase, and then they are just depending on what is automatically spontaneously released into the aqueous phase. Therefore, you can see here that a strain able to degrade this pollutant was phenanthrene. Yes, can degrade the pollutant, but linearly, and if you measure this rate, is exactly the same as the rate of partitioning of the chemical into the water phase. But when you have fungal biofilms on the interface, bacteria can even work better and then access to this interface available chemical, then taking the compound directly at the interface. Even this could be enhanced by different nutrients. Okay, so this is an example how we can enhance the bacterial growth and the interface and promote biodegradation. Finally, with this well-known phase exchange mechanism, you may have heard about biosurfactants, that are produced by bacteria, by plants, that can enhance the dissolution and solubilize or pseudo-solubilize chemicals that are sorbed. I wanted to show you today one example we did some time ago with rhamnolipids that perhaps are the best known biosurfactants produced by bacteria. They are very well studied, but our specific study was what is happening when we place them in difficult situations, like aged chemicals, chemicals that are sorbed to the soil for a long time, and then we wanted to see maybe for only 60 days we aged the chemical and see if the rhamnolipid was able to help bacteria to mineralize pyrin. So in this case, you can see that the mineralization rate and the fast degrading fraction were changed even with the H chemical. With the H chemical, the biosurfactant enhanced the mineralization rate, although without aging, you could see very clearly the effect of biosurfactant was very easy, but when the chemical was inside the aggregate, then was different, but was even able to enhance this process. Statistically, we measured the enhanced rate and also the slightly higher fast dissolving fraction. So in this case also, we wanted to test that in a difficult situation, in a slowly dissolving chemical, this is where we want to use biosurfactants, because we don't want to create problems. We don't want to enhance solubilization at a rate that microbes cannot resist, and then we create a problem. So the idea is really to apply the biosurfactants in the slow dissolving fraction, in the later rate, where you don't create a problem, you don't enhance the risk, but the opposite, you help bacteria to even go down with the residual levels. This was the philosophy we wanted to test. Now, what we do with all these experiments, how we can apply that in a real world scenario. All these have been model systems that help us really in understanding process, but in bioremediation, well, you have the soil, you have a situation that you have to handle. So the idea is really how to connect these engineering components, how we can also help with this situation where, for example, in bioremediation, we may have an increase in risk initially, and we don't want that. We want to go down. Later on, we will see how we go to the second phase. So this is, for example, an experiment we wanted to interrogate what is this situation, no? That we have initial easy carbon pollutant released very quickly, and then we applied C14 labeled chemical, was pyrin, was a 4-ring chemical in greenhouse experiments, where we measure the concentration of pyrin equivalents, which are labeled with C14, in the leachates of these pots that were incubated in greenhouse. So we could see that this was a clean sand system, where we had the pyrin in the bottom layer, and then without bacteria, we could see that even the plant alone, the sunflower, could even degrade the chemical and produce a very high level of pyrin equivalents that were well above the solubility of pyrin in water, which is about 0.1 milligram per liter. So you can see here the production of metabolites by the plant that could be a risk also, no? Could be a risk that we reduce if we applied in the bottom a sorbent phase like biochar, that we then neutralize this risk produced by the biodegradation process. In a second test, we wanted to check, this was also one part of a bigger project where we really examine how bacteria can be mobilized by the sunflower roots to this distant pollutant source. So we applied planting and inoculation with this Pseudomonas putida strain, which is able to co-metabolize pyrin, and also seeing that we could see an increase in production of pyrin metabolized in the leachates of the pots in combination of plants and inoculation. So without bacteria, we didn't see this high concentration of metabolites produced by the plant, and we attribute that by the effect of bacteria, of inoculated bacteria, in preventing the stress response by plants and causing this biodegradation. In any case, we could see that again by the application of this biochar layer in the bottom of the pots, we could decrease the risk of biodegradation. So having a whole set of inoculated system with plants that were working to enhance biodegradation, but not creating this risk. So the second example was what to do also once we have solved this at a high risk, how we manage to go down in risk. So we try to combine the knowledge we had on these biosurfactants and also plants and see how we can apply the biosurfactants not at the start of the process because we don't want to create more problems. The idea is also try to enhance the action with the slowly dissolving chemicals that are remaining after the first degradation phase but are still above the legal standards. So we did another experiment in greenhouse, in this case where with a creosote polluted soil that was rich in polycyclic aromatic hydrocarbons, measuring at the same time the evolution of the total concentrations of the PAHs and the bioavailable concentrations of the PAHs by doing the 10x extraction that gives you the right measure of the bioavailable fraction. I will come to that later, what is the meaning. So here is the situation where you can see the evolution of the total concentration of PAHs in planted and unplanted pots. The arrow means the time point in which we added the biosurfactants really to enhance bioavailability to promote biodegradation. At the right side, you can see the evolution of bioavailability measured in parallel to the total concentrations. You can see here a very good example that the evolution of bioavailability is giving you more information in addition to the total concentration of the chemicals. Here you can see the bioavailable fraction going down, and again when you add the biosurfactant is increasing, okay? So significantly by the addition of this biosurfactant. What is not happening in the unplanted pots. So this means that biosurfactants action were enhanced by the development of the planted sunflowers, so they in combination enhance the bioavailability of the heavy molecular waste, the high molecular waste PAHs. Although you can see therefore that this enhancement in bioavailability was not followed by enhanced dissipation of the chemicals. So we selected these soil samples and treated them through shaking and biostimulation to test if this enhancement in bioavailability at some point, if you modify the ecological conditions different to the greenhouse experiment, you could get a decrease in residual concentrations. And this is the result of this experiment, where you can see in this slurry experiment of these samples taken from the greenhouse, where we added a C14 labeled pyrene to monitor the biodegradation process. At the end of this treatment, we measured the residual concentration of pyrene in the soil. There you could see, therefore, an enhanced biodegradation by this addition of biosurfactants and plants. You could see, therefore, that the higher bioavailability detected by 10x extraction was giving you 20% of bioavailable pyrene as compared with only the soil that had 4 milligrams. This is in milligram. No, this is in percentage. So these different 10x extractabilities were correlated well with degradation observed in the slurry treatments. So this was only occurring in the combined system of plants and biosurfactant. The initial concentration of the polluted soil was about 60 milligrams per kilogram. Therefore, you can see that we could go even 0.4 milligrams per kilogram, so showing the success of the bioremediation treatment here. This could happen also with benzoapyrene, which is a 5-ring polycyclic aromatic hydrocarbon, and therefore we could see that combining biosurfactants and plant could decrease the concentration for 4.1 milligram to below 1 milligram, which is the limit. So this is probably I am extending too much. Now I have the second part on regulation, but I will try to be brief. So this was the basic research. We wanted to understand a bit the scientific part of bioavailability, but what if we like to use this for regulation, okay? So during the previous 35 minutes, we were here. All type of complications are valid as long as they help us to understand the bioavailability. But now, when you need to talk about how to use this, people that have to make decisions have to say this is a polluted site, this is not a polluted site, then you have to be sharp. And sometimes you have to make simplifications which are valid in this context. Okay? So therefore, we change a bit the mind and then see how we can help with that. Okay? So one way of helping, only one way, is with science you can provide methods to measure bioavailability. Okay? This doesn't mean that you will solve the issue, because there are other parts of the regulation that are financial aspects, social aspects, or all of these that are not scientific, but this is the scientific part where you can provide a method to measure, like this one here, this ISO standard that use our original effort in this ISANTI paper that we provided this simplification. This ISO standard can be used today to measure bioavailability of hydrophobic chemicals. So maybe you can just consider that the bioavailable fraction is the fraction of the total pollutant that is desorbing from the soil after 20 hours. That's it. And then just by simply using that, you can measure this type of desorption by Tinax. It's a resin that is sequestering all the chemical once it's in the water phase. This is in this ISO method. You can use also cyclodestrins that are having a similar effect. Therefore, in this way, you can determine the bioavailable fraction. By this way, you can add more realistic assessment to your assessment based on total concentration, which is today not enough. So total concentrations are giving you a good measure, but you need also more realism, for example, with exposure, therefore with these bioavailability measurements. You can also measure what is in the equilibrium in the water phase by passive sampling, which is also a very useful technique. It's different, I will come to that later. So in this way, you can really help regulation by also providing this way of measuring. So this is this ISO method was built in a retrospective risk assessment scenario. This means for legacy pollutants, the industrial sites, etc. But the idea is also, and we also propose that could be used in prospective risk assessment. Means for new chemicals that need to be regulated, need to be registered, you need to provide a dossier of really persistence, and etc., and also bioavailability. So the idea was really, we published that together with a group from the industry, trying to also to promote this in a prospective risk assessment. So you could combine bioavailability measurements with, for example, OCDE methods for measuring persistence, in which you can measure total concentration and bioavailable concentration. And this is, as you have seen, giving you more information of what is happening really in the biodegradation process. Okay, so then we can find these two schools of measuring bioavailability. No? I mentioned this desorption extraction, this passive sampling. Where are these based? On which concepts? And we can see, for example, an analogy of the total pollutant mass on an iceberg. No? For example, an iceberg where is floating in the water phase, and most of the pollutant is below the water phase, is sorbed to the sediment, and then what you measure in this equilibrium situation is the freely dissolved concentration. And this is a minor part of the total mass, but this giving you the right measure of bioavailability, because this equilibrium concentration can be applied in all this partitioning modeling, all this ecotoxicological knowledge that are absorbing very well this type of measurements. This is a sound measure of bioavailability. The thing is that not always you face equilibrium situation in nature. Even, for example, in biodegradation processes, like the right side of this picture, there is a process removing the chemical exposed into the water phase, like this sun melting the upper part of the iceberg. So really what you have to measure is what is just below the water phase. And this is the fast desorbing fraction. You can measure that with a desorption method that take all this most dangerous part of the chemical, which is giving you the risk. This is also there. This is also, but it's less risky. So it's in the bottom, so very hardly creating you a risk. What you have to know is what is the mass of really the risk part of the chemical that is giving you. So this is the situation. The passive sampling methods are well described. You can find in this paper, Nature Protocols. It's not standardized, but still there are strong moves towards a standardization of these passive sampling techniques that are very well developed in connection to bioavailability. That's the point. So you can measure many things by passive sampling, but passive sampling for bioavailability, you need this third phase, which is the soil, the sediment, and there you have to make this type of application in a bioavailability concept. In the desorption extraction, you have seen this method, this ISO method, is ready there. So you can use that ready to measure bioavailability by this Tenax extraction, cyclodextrin extraction, that can give you the right measure of risk of the chemicals. So these are today complementary. You can use both of them depending on the situation. For us, we have been reasonably successful with the desorption extraction, with Tenax extraction in terms of bioremediation. You have seen some data. So the problem maybe with the passive sampling is that you need weeks to really have the right equilibrium. And this is a problem if you need, for example, if you are in a commercial lab and you need fast measurements and so on, that with the desorption extraction you have 20 hours and then that's it. You have a measure, you total extractable and Tenax extractable. So these are important parts of the standardization part, and maybe there's a good chance that they are used now that we have this ISO standard for measuring bioavailability. This could be done in a risk assessment scenario. Once you detect the total concentrations are giving you potential problem, then a second stage of assessment, you can use this type of measurements. This is, some countries use this approach already, some specific, but in general, there is still a lack of application in different countries. In the European scenario, I will show you some examples. As I told you, very personal view of how I perceive bioavailability fits in this type of developments, and also, for example, here in this global assessment, not only in European, a global stage by the United Nations Environment Program, where really you can find a place for bioavailability. They have detected, now you can see a stream of thinking that soil is important. So we have been environmental issues on water, atmosphere, but soil now is becoming an issue. All these crises that we have around, the war we have in Europe, and so we have to produce food, and soil is becoming the attention, which is a good idea. And then the idea is really that these policies consider aspects that where bioavailability can help, like, for example, this bioremediation, this nature-based management that have really a good result in terms of risk reduction by attacking bioavailability, and also this could help also other countries where this knowledge is not easily available. And a nice surprise that when you look at the glossary of this report, the first word that is in this glossary is bioavailability. So it's a concept that they are considering, is something, they don't say how to use it, but here we are to help them. And the idea is that we have this knowledge, and this is an important issue in soil pollution. Soil pollution, well, and remediation for sure. So in this report, you can see, therefore, a situation. They make really a detailed analysis on all the soil pollution in different globe areas. And here in Europe, you can see that about 600,000 of potentially contaminated sites by industrial activities, agriculture. These blocks, the size of these blocks are relative to the amounts of polluted sites, potentially polluted sites in Europe, and they provide also different regions like North America. They have also different blocks in North America. The mining is becoming more important, but again, the industrial activities is giving you most of the soil pollution. And if you look at detail of the type of pollutants, the red highlighted pollutants have a bioavailability phase. And here I have included also trace elements. So all these red chemicals, pesticides, DDT, PCBs, they have a bioavailability issue for sure. Sometimes they are hydrophobic. So really, you improve the assessment by considering bioavailability, like military chemicals, etc. So, and you can treat also these chemicals through nature-based remediation. We did this review recently also, where really you can view all these nature-based strategies by reducing bioavailability. So not reducing the total concentration. So you have to change the paradigm of treating the total concentration, but they are very useful in reducing the risk by reducing the bioavailable fractions. And it has been so useful in this context of military activities. So maybe I will just go through, and you all can see we are in a sustainable institute. So I promise you there are good connections, very good connections to many of these goals by improving crop production, by improving environmental health. So really, by using bioavailability as a risk tool, risk assessment tool, you can really contribute to many sustainable development goals. So I will go through this very quickly. And also in Europe, I wanted to show you that we have the Green Deal. I don't know if you have heard about that. There is a big political move for the Green Deal that is moving a whole set of policies in Europe. And here is I showed you the European soil strategy, which is a strong move in soil health quality in Europe, which is touching all these Green Deal policies that are zero pollution action, the chemicals strategy, etc. Okay, so and there you find a good place for bioavailability, really. So you can really improve the information you provide on the soil, on polluted soil, if you incorporate risk assessment on the basis of bioavailability. And also the zero action pollution plan that we have also in Europe from this Green Deal strategy, where the strategy is to have minimization of exposure. Then you, when you attack bioavailability, sure, you minimize exposure, and also you can improve the treatment through this biological, nature-based approach, and enhance this policy. So as a conclusions, I think I am okay with time. So total concentrations are useful, but you can improve that assessment with bioavailability. These approaches are fully in line what is the currently sustainability goals and the European Green Deal policies, and for the basic point of view, you can really operate on these low pollutant exchanges by different mechanisms to enhance bioremediation. And before I finish, I wanted to invite you next year to Seville, my home city, where we will organize next year the CTAC conference, and you are mostly welcome to come. So you can get the details also from Professor Ghoshal, where we will be around 3,000 people around. So it will be a good context to see really environmental science in the European side, but is also attending globally. No? So thank you very much for your attention, and for those who like to come, I look forward to see you. So thank you.